Chapter 10, Muscle Tissue, Part 6. All right, moving on to cardiac muscle tissue and smooth muscle tissue. Cardiac muscle cells are much shorter than skeletal muscle fibers, and they often branch so that they have little, branches coming off of them. They're shorter, thicker, they branch. Cardiac muscle cells tend to have only one, sometimes two, but usually one nucleus per cell. They do possess the striations, though. So they do have the striations, they possess the myofibrils, the sarcomeres, and they use a lot of aerobic respiration. So lots and lots of mitochondria within each cardiac muscle cell. The cardiac muscle cells are wrapped around by the endomysium, but they lack epimysium and perimysium. So they only have the endomysium around each individual cell. Cardiac muscle cells do possess protein structures called intercalated discs. Intercalated discs form where two cardiac muscle cells are joining together. So this is a type of junction. And the intercalated disc is composed of many desmosomes, which allow for strong connections between the two cells to hold them together against forces being generated by the contracting of the heart. And there's also a lot of gap junctions forming tunnels between the two cells so that fluids and ions can move from one cell to the next cell. So those are the junctions found at the intercalated discs. Cardiac muscle tissue is also autorhythmic pacemaker. They are able to contract on their own without a stimulus from nerves. So the autorhythmic pacemaker is causing repetitive rhythmic heartbeats. So it's causing the constant beating of the heart and is independent of the brain. So it itself is what stimulates the cardiac muscle cells. However, how slow or fast or how hard the heart beats is controlled by the brain via the autonomic nervous system. So the beating is caused by the heart itself, but the speed and strength is determined by the nervous system. All right, smooth muscle tissue. Smooth muscle tissue is found in the hollow organs of the body, except the heart. It's found in blood vessels to regulate blood pressure, the bronchioles of the respiratory system to control air movement, the small and large intestines, and a lot of in the heart and the stomach for mixing and propelling materials, the urinary bladder, the ureters for the urinary system, the uterus, very important for childbirth, and many, many others. So almost all the hollow organs of the body have smooth muscle tissue in their walls. Smooth muscle tissue is sort of fusiform in shape, so it's wide in the middle where the nucleus is and then sort of tapers off at the ends. This allows the cells to be really closely packed together. Smooth muscle cells each have one nucleus. They are much smaller than skeletal muscle fibers. And they have a wrapping of endomysium around each cell, but they do not have perimysium or epimysium. So again, only endomysium around each individual cell. So another important characteristic is that smooth muscle cells do not have striations. That's why they're called smooth. We do not see that striated pattern. Their sarcolemma has a lot of calcium ion channels along its length. There are few to no transverse tubules, and there is little to no sarcoplasmic reticulum. So the primary source of calcium ions for smooth muscle tissue is actually outside of the cell. And of course, as I said, no striations. So instead of having myofibrils, the arrangement of the proteins that are sort of like the sarcomeres is more dispersed throughout the cell and connected directly to the cytoskeleton. So again, extensive cytoskeleton within the smooth muscle cell, lots of specialized intermediate filaments. Then there are points where the intermediate filaments interact with the sarcoplasm, and that is referred to as dense bodies. So dense bodies are places where the cytoskeleton is making contact with the sarcoplasm. And then there are also dense plaques. Dense plaques are places along the length of the intermediate filament where it is anchoring with the neighboring dense plaques. So it's where they're interconnecting with each other. So contractile proteins. Contractile proteins are still present. We still have thick filaments and thin filaments. And so as you can see, thick filaments are still attaching to the thin filaments, connecting between the dense bodies attaching to the intermediate filaments attaching by dense bodies to the sarcolemma. So the myofilaments of the smooth muscle include myosin as the thick filaments and actin and tropomyosin on the thin filaments, but there's no troponin in the thin filaments. And there are heads, myosin heads, all along the length of the thick filaments. This picture is actually wrong. There should be myosin heads shown here in the center to allow for additional cross bridges to form. So instead of troponin, we have these proteins out in the sarcoplasm. We have calmodulin. Calmodulin will bind to calcium to form the calcium calmodulin complex. When that happens, it will activate myosin light chain kinase. Myosin light chain kinase will then phosphorylate the myosin heads of the thick filaments. That then stimulates the ATPase activity so that the myosins are activated. So again, calcium ion concentrations increase, bind to calmodulin. Calmodulin changes into calcium calmodulin complex. That activates myosin light chain kinases, which then phosphorylate the myosins in the thick filaments, thereby activating them so that they can begin the contraction cycle. So steps of contraction, again, stimulation from a nerve impulse, from some other signal, allows calcium ions to enter through voltage-gated calcium ion channels. The calcium binds to the calmodulin complex, which then activates the MLK, which then activates the myosins by phosphorylating the myosins, allowing it to do the power stroke and reset to do more power strokes again and again and again. And so we get the repetitive release and attachment of myosins to actin, pulling the dense bodies closer to each other. So the filaments are sliding past each other, the thin filaments toward the middle of the thick filaments. Dense bodies are being pulled closer and closer to each other. This is then pulling on the intermediate filaments of the cytoskeleton, which is causing the dense bodies on the cell membrane to get pulled toward each other. And this leads to an overall shortening of the cell. The cell contracts. In relaxation, basically, there is a myosin light chain phosphatase. Phosphatases remove phosphates. So this enzyme, when activated, will remove the phosphate on the myosin, causing it to, once again, turn off to inactivate its ATPase so that it cannot go into the contraction cycle. But wait, there's more. We also need to talk about how the smooth muscle tissue contracts. Well, it turns out it has a longer latent period, so it takes longer for the stimulus to lead to the contraction. And this has to do with, well, the more complicated steps in getting the myosin activated because of that phosphorylation step. And then the duration of the contraction is longer than in skeletal muscle cells. Because of the slowness of the calcium pumps and dephosphorylation of the myosin heads. Basically, it takes longer for contractions to occur in our smooth muscle tissue, and that is good and that is bad. Basically, it's fine for our visceral organs. It allows for them to maintain a continuous tone and to contract for longer periods of time because everything's just a little slower. They are also more fatigue resistant because they are going slowly. There's less energy requirements for smooth muscle tissue, so they need less ATP and can usually get what they need through aerobic cellular respiration. And so it's actually pretty difficult to fatigue your organs inside your body. Can happen, but it's much more difficult. Their overall curve for the length tension curve is more broader because, again, they have the myosin heads present along the full length of the thick filament, and they aren't limited by Z discs. So there is a longer range of stretched and contracted sizes of the smooth muscle tissue that can still allow for force to be generated. So they contract more forcefully even when compressed to half their length or stretched to double their length. So, and this is important because, some of our organs stretch a lot. So the urinary bladder, for instance, gets extremely stretched as it fills. And so that's going to stretch the smooth muscle tissue in its walls, but we still need the urinary bladder to be able to contract to get rid of the urine. So, which protein activates myosin in smooth muscle fibers? And the answer is myosin light chain kinases. Again, kinases add the phosphate group, phosphatases remove it. All right, controlling the smooth muscle tissues is controlled by the autonomic nervous system. It's involuntarily controlled. We do not consciously have to make our intestines contract or our stomach to contract or our blood vessels to contract. And it responds to a neurotransmitter. Surprise, surprise, it's acetylcholine. So these smooth muscle tissues will contract in response to acetylcholine being released. And also, they will relax in response to norepinephrine, the type of adrenaline being released. So when we get excited by adrenaline, it actually relaxes a lot of our inner hollow organs, like the digestive tract. Again, contraction can also respond to stretch, so that if it's it will respond to if it's continuously stretched. If the stretch is prolonged for too long, it will contract automatically. This allows the smooth muscle tissue to prevent itself from being overstretched. First it'll contract, then it'll relax. Other things that can affect smooth muscle tissue contractions, lots of things. Hormones, pH, oxygen levels, carbon dioxide levels, drugs, lots of things. So for instance, oxytocin is a hormone that can stimulate the contraction of smooth muscle cells in the uterus. So certain smooth muscle tissues respond to different things in different ways. Categories. We have multifunctional smooth muscle tissue. Multifunctional smooth muscle tissue is where each individual smooth muscle cell has its own connection to a synaptic bulb of a autonomic motor neuron. So iris and ciliary muscles of the eye, erector pili muscles of the skin, airway passageways. We end up with motor units that will all contract together when the signal arrives by the specific autonomic motor neuron. Great for more units being stimulated, stronger contractions. In single unit smooth muscle tissue, the neuron is modified. It doesn't actually make these neuromuscular junctions. Instead, it's going to release its neurotransmitters and have them bind to smooth muscle cells that are nearby. And then those muscles will contract, and the signal will pass on to neighboring smooth muscle cells via the gap junctions that are between adjacent smooth muscle cells. So, for instance, the walls in the digestive, urinary, and reproductive tracts, some of the respiratory tract, most blood vessels, single unit. The signal spreads more slowly from one cell to the next cell via gap junctions. So basically, like I said, here is the neuron. It has synaptic vesicles within these varicosities that will release the neurotransmitter that will lead to the contraction. So it releases the acetylcholine. And then slowly through gap junctions, it'll spread to the other smooth muscle cells to get the entire tissue contracting. And that is it for this chapter.